And we're rolling. This is not going to last particularly long, but I wanted to talk you guys through the uh, initial steps of the lab, or not the lab, the final that I want you folks to take. And Jackwagon over here will give me a hand, I'm sure. Hello, you cat. So let me. All right, so let's cut over here. This is the ncbi.nlm.nih.gov is the National Center for Biotechnology Information. I believe you folks used it before when I had you do another phylogenetics assignment. But here I want you to actually do some searching and find some sequences and put them together for analysis. And then most of the exam is going to be devoted to trying to figure out what the heck it means. Drawing the tree is the easy part. So if we come down here and we hit taxonomy over on the left, there's the taxonomy database. And up here at the top, what I've given you is Echino, PHTHIRIUS, horridus, which is actually in one of the presentations. It happens to be a louse that is a parasite of seals. And I found this really weird because there's not an awful lot of insects in marine environments. And you would predict that an ectoparasite would be at a distinct disadvantage on a critter that, hauls out for part of the time, but spends a great deal of time, swimming in cold ocean water. But there you go. There's a whole group of lice that's actually pretty well adapted to life on marine mammals. This was cool. I actually found a website with some researchers who worked with some sea lions that haul out on barges within a city. I think it's in Argentina. And they were able to invent these very fine combs with extremely long handles, and were actually able to reach out and scratch these sea lions, who actually got to enjoy it very much. And they ended up sampling, it's not Echinoptyrus, I think it's Antarctoptyrus, but they ended up sampling these lice. Kind of a clever way to study these comparatively safely. So if we search on Echinoptyrus horridus, wakata, we click over here on nucleotide over on the left. And this is what GenBank, the database of DNA sequences, has. They've got 11 different molecular, well, DNA anyway, sequences for Echinoptyrus horridus done by previous researchers. So we've got a bunch of them. If you scroll down here, you get this. This is Echinoptyrus horridus 18S small subunit ribosomal RNA. If you forgot, you should know what ribosomes are. They're the things that make proteins, and they're made of protein themselves, but they're also made of specialized RNAs. And there are 4 separate RNA molecules, and they're called the 28S, the 18S, and then I think the 5.8S and the 5S, if memory serves. So if you just click on this, you get a whole lot of information as to, how this is classified, where it was published. This is published in BMC Evolutionary Biology. If you see that, you get the authors, you get the title, you get all of this, you get some codes, and there's your DNA. That's important just for keeping track of what you're doing. But if you want to do the analysis, what you want is called FASTA format. So we go up to the top and we click on FASTA, and this is what we get. And it's nothing but a title. This number right there is just the accession number. It's the catalog number for the data. And all you have to really do is copy this, and you can paste it into a Word document or whatever, whatever word processing document you want to use. And so there's the lab handout, and there it is right there. It's quite a lot of A's, T's, G's, and C's. And from our previous page over here, over on the right, there's a link to running BLAST. And BLAST is a program that will comb through this enormous database and pick out highly similar sequences. So right up here, we've entered, that's the accession number that you saw. That's that catalog number of the sequence. You can scroll through this. You don't really need to do much with these default settings, although I guess you could mess with them if you really wanted to. Oh, look, notice on the right, they've got an entire database dedicated to coronavirus sequences. A lot of people are using this right now. And just go down here and click on BLAST. And we can put this DNA sequence on BLAST. In fact, there's actually quite a lot of things I would like to put on BLAST. And it updates the page periodically. And boom, we've got our results. So scroll down, and we have 100 sequences that are similar to Echinoptherius horridus. The one at the top, by the way, is the one that we started with at the beginning. That's obviously 100% identical to itself, and that's not particularly interesting. But look, here we've got Lineagnathus ovilius. I don't happen to know. Actually, I do. I think that's one of the lice that can infect dogs. Although, Nick, you were telling me that you don't see true lice on dogs very often. But that right there, you can see, if you look at that percent identity column, that one's 97.03% identical. So if we click on it, we get that. That's our, up at the top, that's our original sequence, and at the bottom, that's the new one, and you can see how many matches there are. But really, we can just click on this, and we get Lineagnathus ovilius. Click on FASTA, and we get that, and we can copy that and just paste that right into a Word document or whatever processor you're using. We can use the same, we can paste it right into the same file where you pasted that Echinoptherius horridus file. And so all we have to do is, copy this and leave a space and then paste it right in. And it'll look like that. All right, and then we can, of course, go back. Okay, there's our BLAST results. Oh, wait, that is our job. Okay, I just want the descriptions there. Okay, and then the third, if we want to, Lineagnathus africanus, we could do the same. The fourth right there is Hematopinus tuberculatus. I don't even know what that is, but click right there. there it is in GenBank format, and we can click on FASTA right here and copy that and paste it, and so on. And you could keep doing this as long as you wanted to, which I assume is not going to be immensely long. But what I've asked you to do is to pick 12 of the sequences that you get from that blast search, copy them in FASTA format, paste them into a document, and you probably need to save it as text, I think, .txt. And once you save that, you go to www.phylogeny.fr, which is what you did before. And we go to phylogeny analysis, and we can go ahead and just do one click. And you could either cut and paste your sequences here, or you could choose the file and upload it. And the thing will go crunch, 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 and spit out a tree. And most of this lab is just going to be involved with, come on, Wagoner. Most of this lab is just going to be involved with trying to figure out what the tree means. But I just got interested in the fact that marine mammals have lice, and that just kind of struck me as beautifully weird. And I wanted to see what you folks would make of it, have you do a phylogenetic analysis, and see what you can conclude about the origins of these, and then see if you can think like a parasitologist a bit and come up with something interesting to say as to what happens when a taxon makes a transition, like going from land to water. Either it would pick up new parasites from unrelated organisms that are already in the water, or it might bring its land parasites with it. One way or another, that's going to improve a lot of selective pressure on the parasites. And I just wanted to throw out, what do you think would happen? And we'll see what we come up with. Okay. Any questions? Was that horribly confusing?
Yes. No.
No, you're good.
No.
Okay. Do I need to, are there any details I can go back over?
Can you, okay, so if we email you questions about this, will you respond to our emails?
Yes.
Promise?
Just no sloma.
Okay. I'm
All
holding
right.
to it.
Okay. I'm going to go ahead and stop recording if that's all right. All right. So we'll stop recording here. Hope this helps people.